Hello dear friends and a warm welcome. In this video you will get important multiple choice questions on microbiology and applied microbiology. This is the question paper of MSc Entrance Exam of Applied Microbiology conducted by Banaras Hindu University. Now this test is conducted by the NTA under the CUET PG exam. This question paper contains a total of 150 questions, out of which in this video you will get the explanation of the first 50 questions. So watch this video till end because it is very important for you if you are preparing for the UGC CSIR NET, ICAR-NET, ICMR-NET, GATE exam, for the CUET PG exam or any of the entrance exam for admission to the MSc course. Myself, Dr. Tripti Manik Ahuja. I am your tutor, guide, and mentor for the subject Biology, Life Sciences, Microbiology, and Biological Sciences. In the description box, you will find there is a playlist, and in the I button here, the link is given for the similar videos which will help you in the preparation of your exams. So now let's get started. This question paper contains total of 150 questions, and we are going to discuss the first 50 questions. These are multiple choice questions, so I suggest you to take out a paper and pen, and I will read the question and then the 4 options, out of which one option is correct. The option which you feel is correct you can write on the paper, then I will tell you the correct answer. You can match your answer with that of the correct answer. And if you want explanation, then you can write in the comment section. Although I will give you some explanation here also. And if you disagree with any of the answer, again you can write in the comments. I will reply back to each and every comment. Now question number 1 says, on what evidence did Aristotle hypothesize on spontaneous generation? 1. Animal cues. 2. Experiments with broth. 3. His belief in magic. 4. Observing living things seemingly arise from non-living matter. Aristotle was a believer of the spontaneous generation, according to which life arises from non-living matter. This non-living matter, he called it pneuma. So according to these options, the option number 4 is correct. Next, John Snow made what important contribution to microbiology? 1. Discovered the vaccine to cure cholera. 2. Was an English physician. 3. Mapped the cholera cases in London and found their source. 4. Discovered how to stain bacteria so they could be studied. John Snow was a famous epidemiologist. He was also called father of epidemiology. He disabled the handle of hand pump, the hand pump which was called Broad Street pump. And then the cases of cholera reduced. So the option 3 is correct. Question number 3. Who developed postulates to demonstrate that microorganism caused disease? 1. Semmelweis, 2. Metchnikoff, 3. Koch, and 4. Chamberlain. Out of this, the option 3, Koch, is correct. The full name is Robert Koch, also known as father of medical microbiology. He gave the famous 4 Koch postulates, which are important in medical microbiology. Question number 4. Christian Graham solved what important problem faced by microbiologists? 1. How to kill gram-positive and gram-negative microbiology. 2. How to view microbes and distinguish between 2 main types. 3. How to prevent the spread of infectious disease. 4. How to make microbes more attractive through the use of colorful dyes. I hope you remember the Gram staining. Gram staining is a differential staining given by Christian Graham, because of which the bacteria, they can stain either the purple or they stain the pink. If they are staining purple, they are called gram positive, and pink ones are called gram negative. The staining difference is because of difference in the cell wall composition of bacteria. So here the option which is correct is the Option 2. How to view microbes and distinguish between two main types. I hope you are getting the correct answers. Question 5. Endospores are 1. Reproductive structures of fungus, 2. Living vegetative cells of bacteria, 3. Dormant structures produced by stressed bacteria, 4. A type of vaccine. So out of this, the option number 3, dormant structures produced by stressed bacteria, is correct. The stress bacteria means the bacteria which are having the unfavorable condition of dryness, high temperature, or loss of the nutrients. Question number 6. Genetic recombination occurs in bacteria through the transfer of 1 pili, 2 plasmid, 3 endospore, 4 autospore. The correct answer is the option 2, plasmids, and this process is called conjugation. So I hope your answer is correct. Question number 7. Any process that results in the integration of new combination of genes together in a single cell is 1. Genetic recombination, 2. Fusion, 3. Genetic fission, 4. Allelotyping. The option 1, genetic recombination, is correct. Genetic recombination in bacteria occurs by 3 processes. One is conjugation, the other 2 is transduction and transformation. Question 8. Gene transfer in bacteria by transformation has the following characteristic. The option 1, a majority of donor genes are transferred. 2. Involves a plasmid. 3. It depends on phage infection of the recipient cell. 4. It can be carried out using free DNA extracted from the donor. So here the option which is correct is the option 4. I hope you remember the Griffith experiment which was carried in the Diplococcus pneumoniae, and it involved the transfer of the factor called the S factor. Question 9. A transducing phage 1. Contains only viral DNA. 2. May contain viral and bacterial DNA. 3. Is sensitive to DNAs. 4. Can never transfer extra chromosomal genes. So here the option which is correct is that it may contain both the viral and bacterial DNA. Now the question number 10. It is also related to bacterial genetics. Which of the following statement is false concerning a meeting between F positive and F negative cell? Note the false statement you have to pick out. 1. The F negative cell is converted to an F positive cell. 2. F positive cell is converted to an F negative cell. 3. Chromosomal genes are rarely transferred. 4. Cell to cell contact is always necessary. So here the option which is correct is the option 2. The F positive cell is converted to the F negative cell. So when there is the transfer of the F factor, before that the F factor or the F plasmid gets replicated. So when the factor gets transferred from the donor to recipient, it does not get converted to the F negative cell. Question number 11. A viroid is made of 1 protein only. 2. Capsid and genetic material. 3. Genetic material only. 4. A capsid only. Now what are viroids? Viroids, these are the virus-like particles. Particularly they contain the single strand RNA, but there is no protein coat. So the option 3, this is correct. Now moving to the next question, the question number 12. When a virus invades a bacterium, gets that bacterial cell to make more copies of the virus that eventually burst out of and kill the same bacterial cell. This is called 1. Lysogeny, 2. Lytic replication, 3. Murder, 4. Prion. So here the answer which is correct is the option 3. This is called the lytic cycle. Question number 13. Which of the following is not an acellular infectious agent? So note down, it is asked not an acellular. The option 1 viroid, 2 is bacteria, 3 is virus, 4 is prion. So it is asking not an acellular. That means which one is cellular. So out of this, the viroid, virus, prions, these are the virus or the virus-like materials which don't have a cellular organization. But bacteria, these have the organization like a cell. So the option number 2 is correct. Okay. So next question number 14. When a bacteriophage is integrated into a cellular genome, it is called a 1. Virulent virus, 2. Lytic virus, 3. Prophage, 4. Transducing virus. Here the option which is correct is the prophage. So this is happening in the virus which undergoes the lysogenic life cycle. Question number 15. The infectious substance of prion is 1. Protein, 2. Glycophosphate, 3. RNA and 4 is DNA. So what does the prion stands for? Prion stands for the proteinaceous infectious particle. They have only the protein. So the option number 1, protein, is correct here. Next question is question number 16. The only structural pattern that has been found among isometric viruses is 1. Icosahedral, 2. Spherical, 3. Helical and 4 is tetrahedral. So now the structure patterns means the symmetry which is found in the isometric viruses. The option 1, icosahedral, is correct. The icosahedral symmetry, here there are 20 sides and each side is an equilateral triangle. Question number 17. This is a bouncer question, a very simple, very easy question for all. Viruses are considered to be 1. Non-living, 2. Primitive precursors of bacteria, 3. A link between life and non-life, 4. Primitive organism. So here the option 3 is correct. I'm sure you have got the correct answer. So these are the connecting link between life and non-life. Some characteristics are living and some are non-living. Question number 18. Which of the following is the arrangement of organisms into groups or taxa? 1. Nomenclature, 2. Identification, 3. Systemics and 4 is the classification. The correct option is the 4, classification. This is the orderly arrangement and characterization of all the living organisms. Question 19. Deoxyribose is 1. C5H10O5, 2. C5H10O4, 3. C6H12O6, 4. C5H12O6. Deoxyribose is a pentose sugar which is found in the DNA deoxyribose nucleic acid. It differs from ribose in having an oxygen atom less at the 2 position, and here the correct formula is the 2C5H10O4. Question 20. An amino acid without asymmetrical carbon atom is 1. Histidine, 2. Threonine, 3. Phenylalanine, 4. Glycine. The correct option is the 4, glycine. The glycine has no achiral carbon atom. Now the question number 21. Decreasing order concentration of minerals inside the cell is calcium, potassium, sodium. Option 2, potassium, calcium, sodium. K, NaCa, NaKCa. The option which is correct is the third option, which is the KNaCa. The potassium and the sodium ions, they are present in higher concentration. They are required for the action potential, and the calcium, it is required for the structure organization of the cell, although it is not written, but here we are considering it for a mammalian cell. Question number 22. Which one is a lipid? Stacose, lycopene, leucine, and the sterol. So out of this, the answer which is correct, we have to tell which one is a lipid. So the lipid here is the sterol. These are the animal lipids. Example of such an animal lipid is the cholesterol. Question number 23. Anticodon occurs in 1 DNA, 2 tRNA, 3 mRNA, 4 rRNA. Anticodon, it is present in the transfer RNA, so the option 2 is correct. It is the trinucleotide present at one end of transfer RNA. Question 24. What is a constituent of natural silk? 1 phosphorus, 2 nitrogen, 3 potassium, 4 magnesium. The one which is correct is the nitrogen. The natural silk, it contains the proteins which are the sericin and the fibroin, and all the proteins, they contain the nitrogen element. So the option 2, nitrogen, is correct. Question number 25. Which enzyme can digest the plant proteins? 1 rennin, 2 erepsin, 3 pepsin, 4 all of the above. The correct one is the 3 pepsin. Pepsin can cleave the bond between the tryptophan, tyrosine, and the phenylalanine, which can be found in the plants. Question number 26. Lactate dehydrogenase, LDH. It is an enzyme that takes place in catalysis of pyruvate to lactates. Is an example of isoenzyme, 2 zymogen, 3 coenzyme, and 4 is the apoenzyme. The option 1, isoenzyme, is correct. Isoenzymes, they are slightly different in structure and found in different concentration in the different tissues. Question number 27. Facultative anaerobes are 1 strictly aerobes, 2 aerobes which can respire anaerobically, 3 strictly anaerobes, 4 anaerobes which can respire anaerobically. The option which is correct is the option 2. Actually, these are the aerobes, but they can also respire anaerobically. They produce energy through aerobic respiration and then switching back to the anaerobic respiration. Question number 28. Term bacteria was coined by 1 Pasteur, 2 Koch, 3 Whitaker, 4 Athenberg. The option which is correct is the 4 Athenberg. Athenberg called them bacteria from the Greek bacteria, meaning the little stick. Question number 29. Mycolic acid occurs in the wall of a pathogen which causes 1 diphtheria, 2 tuberculosis, 3 tetanus, 4 cholera. The option 2, tuberculosis, is correct. Tuberculosis causing bacteria is Mycobacterium tuberculosis. Question 30. Which of the following is an antifungal drug? 1 metronidazole, 2 streptomycin, 3 amphotericin B, 4 tobramycin. The option which is correct is the option 3, amphotericin B. The metronidazole is the antiprotozoal, while streptomycin and tobramycin, these are the antibiotics. Amphotericin is antifungal. It acts by the formation of pores, hence causes the leakage in the fungus cell and the death of the fungus. Question 31. Rifampicin acts by inhibiting 1 DNA synthesis, 2 protein synthesis, 3 RNA synthesis, 4 cell wall synthesis. The option 3, RNA synthesis, is correct. Rifampicin inhibits the bacterial RNA polymerase. the enzyme which is responsible for the DNA transcription. Hence, the messenger RNA is not formed. Now, the next question is question number 32. Biodiesel is produced from fats, oil through a process known as 1. Esterification, 2. Hydrolysis, 3. Transesterification, and 4 is condensation. Here the option which is correct is the 3. Transesterification. Fats and oils, they are converted to the biodiesel and glycerine. Question number 33. Prednisolone is antibiotic, 2. Steroid, 3. Enzyme, and 4 is vitamin. The correct option is the option 2. Steroid. These are the corticosteroids which decrease the inflammation while the suppression of the migration of the neutrophils. Question 34. Enzyme immobilization is 1. Conversion of an active enzyme into inactive form. 2. Providing enzyme with protective covering. 3. Changing a soluble enzyme into insoluble state. 4. Changing pH so that the enzyme is not able to carry out its function. The option 2. Providing enzyme with protective covering is correct. The enzymes are fixed on or within a solid substrate or the support. Question 35. Collar and Milston developed biotechnology for 1. Monoclonal antibodies, 2. Myelomas, 3. Immobilization of enzymes, 4. Lymphocytes. The option 1. Monoclonal antibodies is the correct answer. Question number 36. Which of the following is an anion exchanger? 1. DEA cephadex, 2. CEM cephadex, 3. Cellulose, and 4 is starch. The option 1. DEA cephadex is correct. Question 37. Subunits of ribosomes dissociate from each other if magnesium ions greater than 10 to the power minus 2. Number 2. Sodium greater than 10 to the power minus 3. 3. Magnesium ions greater than 10 to the power minus 3 molar. 4. Sodium greater than 10 to the power minus 2 molar. Out of this, none of the option is correct. The ribosomes dissociation is related to the concentration of the magnesium, and when the concentration is lesser than 10 to the power minus 3 mole, then the ribosomes, they dissociate. So here none of the option is correct. All are incorrect. Now question number 38. Gentle treatment of DNA with 1 millimolar HCl leads to 1. Disruption of sugar phosphate backbone. 2. No effect on sugar phosphate backbone. 3. Produce pyrimidine free polynucleotide. 4. No effect on DNA. So here the option 4, no effect on DNA is correct. The lysis buffer which contains the tris HCl lyses the cell, increases the permeability and produces no effect on the DNA. Question number 39. In a peptide nucleic acid PNA, 1. Sugar is replaced by amino acid. 2. Phosphate group is replaced by amino acid. 3. Sugar phosphate backbone is replaced by peptide. 4. Nitrogenous bases are replaced by amino acid. The option 3. Sugar phosphate backbone is replaced by the peptide. This is correct. Question number 40. A solenoid in a chromosome represent 1. 10 nucleosomes per turn, 2. 6 nucleosomes per turn, 3. 8 nucleosomes per turn, 4. 12 nucleosomes per turn. The option which is correct is the option 2. 6 nucleosomes per turn. This is a secondary chromatin structure which helps in the packaging of the eukaryotic DNA into the nucleus. Question number 41. Toroidal state of supercoiling may exist in 1. Negatively supercoiled DNA, 2. Positively supercoiled DNA, 3. Either, 4. None of these. The option which is correct is the option 1. Negatively supercoiled DNA. Then around the nucleosome there are the histone proteins which are 8 in number and DNA is wounded. Question 42. TUS protein is 1. is contrahelicase. 2 inhibits ATP-dependent DNA B helicase activity. 3 blocks progression of replication. 4 all of the above. Here the option 4 all of the above is correct. Thus DNA binding proteins, they arrest the DNA replication, bind to DNA sequences called tersites, and hence forms the replication arrest complexes that block the movement of the DNA replication fork. Question number 43. RNase H has one exonuclease activity for DNA RNA hybrids, endonuclease activity for DNA RNA hybrids, exonuclease activity for RNA RNA hybrids, endonuclease activity for RNA RNA hybrids. The option which is the 2 option is correct. Endonuclease activity for the RNA DNA hybrids. So these are found in all organisms, in the bacterial cells as well as in the archaebacterial cells. The question number 44. SOS response can be activated by 1 purine dimers, 2 misincorporation of bases in DNA, 3 methylated base, 4 quinolone antibiotic induced breaks in DNA. The option 2 is correct, misincorporation of bases in the DNA, which is the damaged DNA. Question number 45. In a type of DNA damage, cyclobutyl ring is formed. It is due to 1 bulging due to insertion, 2 UV irradiation, 3 breaks with deoxyribose ring, 4 breaks at phosphodiester bond. The option 2 UV irradiation is correct. UV irradiation causes the thymine pyrimidine dimer formation. Question number 46. Isoschiromers have the same recognition site, 2 cleavage site, 3 both 1 and 2, 4 origin. The option 3 both 1 and 2 is correct. The restriction endonucleases, they recognize the same DNA and then they make the cut. Question number 47. Homopolymer tailing is useful in 1 ligation of blunt ends, 2 ligation of cohesive ends, 3 ligation of single stranded DNA, 4 none of the above. The option 1 ligation of the blunt ends is correct. Now moving to the question number 48. Insertional inactivation is related to 1 DNA damage and repair, 2 screening of recombinants, 3 termination of DNA replication, 4 translation. The option 2 screening of recombinants is correct. In the insertional inactivation, the fragment of foreign DNA is inserted into a restriction site inside a gene to resist the antibiotics, hence causing a gene to turn non-functional, and this is used for the screening of recombinants. Question number 49. DPN1 is the restriction enzyme which recognizes 1. 2-GC dinucleotides, 2 octanucleotide sequence, 3 hexanucleotide sequence, 4 methylated A residues. Here the correct one is the option 4, which is the methylated A residues. A stands for the adenine residues. These are the type 2 restriction endonucleases. They recognize the methylated adenine rather than the cytosine. Question number 50. The last question of this video is rare cutters for mammalian DNA is ICAR-1, BAM-H1, BSS-H2, then the PST1. So out of this, the correct one is the ICAR-1. This is a rare cutter enzyme which recognizes a sequence which occurs rarely in a genome. One more example is the NOT1. So I hope these questions will help you in the preparation of your exams in the part 2 and in the part 3. The next 100 questions will be discussed. So stay tuned. Take care and best wishes for your exam.